The third area where I think future improvements are going to be possible in the future, we have very, very smart technologies. We have this is a golf buggy that basically sort of runs over the crops, that takes a whole range of measurements. Previously, to take measurements on a crop, we've got to go down there, we have to basically chop them off at the ground surface, bring them back to the lab, measure the biomass, measure the leaf area, measure what's going on. But there are a whole range of non-destructive measurements, a lot of them of which have come from medical research or medical technologies which are now being adopted by crop scientists. At CSIRO we have blimps which are able to sit above crops, which can measure their biomass, can measure what's going on above ground. We have these ground-based systems that do the same thing, which can take measurements of how warm or how cool the plants are, which gives a lot of indication in terms of plant health. And we're starting to be able to take non-destructive measurements of things like protein content, the physiological machinery associated with growth, and so on. So these are technologies where we can start to measure a whole range of characteristics on, let's say, 1,000 different plots or genotypes per day. So significant improvements in this area. That covers increasing yield, increasing biomass production, increasing yield. How can we protect yield and reduce growers' costs as well? A few slides showing different diseases that typically affect wheat plants and what plant breeders typically do. They use molecular markers to pyramid resistance genes. There are 3 different rusts that affect wheat, for example. There's leaf rust, there's stem rust, and there's stripe rust. There are resistance genes that you have to pyramid them together before they are released to farmers. As soon as you start to use this pyramid, resistance pyramid, which protects the crop, and you want to use this as a parent to improve another variety, the whole pyramid just falls apart. So you have to reassemble that pyramid again, and it's a very, very difficult and very chancy thing to do, because the whole pyramid falls apart as soon as you start taking on a breeding exercise. A major opportunity, particularly using GM technologies, I'm less optimistic than Rick about some of the other improvements in GM, but I think this is a major area where GM technology is going to be very important. It's going to be very important by building a cassette, by putting the genes together that are the resistance pyramid and inserting this cassette, this resistance cassette, into different varieties using GM methods. And these should protect the crops for a very, very long period of time, very effectively, and release the breeders to concentrate on other factors such as grain yield. Big opportunity there. Also big opportunities in relation to improving the value of the crop, increasing the value of the crop by producing not just normal starch, which is, say, typically used in producing bread or noodles or whatever it might be, but producing higher value crops for human health. A good example here is using either GM or non-GM technologies, where you increase the amylose content, which is typically low in wheat, increase it higher to produce a resistant starch, which is very effective in terms of human health. The big technology advances, what are they? DNA sequence information is now very cheap. 10 years ago, it cost $6,000 to sequence 1 million bases. Now it's only 9 cents. 9 cents. So massive changes in technologies. There have been big changes in GM technologies. In the last 5 years, we've been able to triple the efficiency of the transformation process for GM technologies just in wheat. So big changes there. Big changes in accurate phenotyping. But the bottleneck in crop science for plant improvement is exactly the same as the bottleneck in human technology, in human science and human health as well. We are able to sequence the entire genome of each of us in this room very, very quickly, very, very cheaply. But this sequence information is of no value whatsoever unless we can relate it to something that's going to have an impact on disease resistance or yield or grain quality. And this is where the bottleneck is. So the phenotypes, what it looks like, is a much greater challenge than the genotypes. And crops are no different to humans in that respect. So I'm wrapping up now. I remind you of the issue that we have ahead of us in terms of the demand for grain and feeding a world population of 9 billion people. So I conclude by saying that global food insecurity is going to be very difficult to avoid. There will be no scientific miracles. Advances are going to be slow and gradual. The big challenge is phenotypic information, not genotypic information, and we must start to build new initiatives to really get us along the road we need to travel on in the future. The implications for Australia, I imagine grain prices will increase as grain becomes, as demand for grain goes up, but input costs are going to increase. There's going to be an increased demand for healthier grains or specialty grains, and Australia, I think, is well set up for that, as well as its clean green grain. I think Australia has got a rare opportunity to provide significant leadership, global leadership in these areas as well, because we do have a very, very strong scientific basis of these. To achieve them, we're going to need significant or important international collaborations and sustained research funding. And I shall leave it there.